Well, thank you very much, Tim, for the nice introduction. I'd like to begin by thanking the organizer for allowing me to present our work here, and thank you all for listening. So before I get into browsing the epigenomes, I'd like to begin by saying that VIZBI has a very special place in my heart. Right? Now, VIZBI is probably the most diverse and dynamic and young meeting that I've ever been to. I mean, you've already seen some really nice talks, and all the slides that you've seen and you're going to see for the rest of the meeting are probably the most, the prettiest slides that you see in all sorts of meetings. But for me, VIZBI has been sort of watching out for my career development. As I told Jim yesterday, this is going to be my opening. Back in 2010, when the first VIZBI meeting was held in Germany, I was lucky enough to be invited and give a talk. And at the time, I just started my own lab. Right? I have a lab of one person, which is myself. I discussed results that sort of work I did as a postdoc at the UCSC Cancer Genomics Browser. So that really marked the opening of my own lab. A year later, I was asked to chair a session visualizing single genomes. I recruited 3 talks, Bob Korn, Brad Bernstein, and Ariz. Bob talked about UCSC Genome Browser. Brad said epigenetics community needs integrative tools and accessible browsers, right? And Ariz discussed the 3D structure of the genome. So these really laid out questions and challenges for people who are interested in visualizing genomic data. So these are the questions I took from this meeting and went back and worked on these problems. And in the second year of my lab, I was able to recruit people. I was able to recruit a very talented postdoc. All the work I'm going to describe today is from this postdoc, and he's here at the meeting, and he has a poster, so I invite you to check out his poster as well. And that's what happened last year. So we didn't go to last year's VIZBI. We were working hard so that we can give a presentation this year. But all these talks are online. That's another really, really nice thing about VIZBI, so I can go back and then check out all these talks. They're still online and alive. Right? So VIZBI is really a special thing for me. It's been watching my career development. So what I'm going to talk about today, I'm mainly going to discuss what is Genome Browser and what's beyond Genome Browser. That's the theme of the session. I'm going to discuss a wish list. What we want Genome Browsers do. We want Genome Browser work like a Google map, right? We want Genome Browser to work as a bioinformatic engine. We want it to not only show us the data, but also do analysis. The challenges outlined from 2 years ago include a very important one, that is visualizing the genome as a linear system. And we also want Genome Browsers to be a community resource. So it's a database. It's a Google engine where you can do your research, you can do your queries, and then get to tap into all sorts of resources out there. So I'm going to discuss these so-called wish lists and how we have been working on to realize this in the context of the WashU Epigenome browser. That's what we've been working on over the past couple of years. And at the end of the talk, I hope we'll be able to expand this wish list. And this is really the goal for me to be here to hear from you, and then to be able to come home with an expanded wish list so that we have more work to do. I already collected one, so we want to visualize variance costs, right? So that's showed up earlier in the slides here. Okay, so just very quick introduction. Probably doesn't need to do so. What is a genome browser? And the best example is the UCSC Genome Browser. Now, I heard Ben mentioned that it's actually very important to hit the sweet spot between, being simple and being sophisticated. And I couldn't agree more. That's exactly why, genome browsers has played a very important role in genomic research. So initially, the browser was developed as a scaffold for the early draft of human sequences. And they took one simple assumption, simple principle, that is the genome is linear. So we can just lay them out and then start aligning stuff to it, right? So you can get the genome in a linear shape. You can put annotations, that CPG island here, gene structures, right? You can have genes aligned to this linear genome, and then you can start aligning lots of other things, conservation. So it's a necessary simplification of the genome, but that allowed us to add all sorts of information to the genomic sequences. Over here, this is where things start to get complicated. When we start annotating the function of the genome, the enhancer state, the transcript, the RNA-seq data that was just asked were shown up as browser tracks on this. And then it really helps us to understand, this part of the genome. What does this part of the genome do, right? And more recently, there's variation tracks, and more recently there's this addition of publications, right? So genome browsers start to tap into the community resources, right? So all the papers, so what you're seeing here, the tracks link to all the papers that talk about this gene, right? So that's all good and nice, except it's oversimplified for a good reason, right? So we know, to navigate the genomic landscape, we have a linear genome here, and we can start to laying on data on top of the genome. And then this list has been expanding just exponentially. And not only that, we just heard about epigenetics, right? So there's a same genome can encode multiple phenotypes. So this really start to grow to a scale that a screen of the computer would not able to expand. And we all know this is the actual genome, right? So how do we do it? Right now we're good at looking at this one screen, one sort of a surface of this data, but the genetic or epigenomic data is really much more complex than this, right? So we want to start sort of tapping into how to navigate this landscape. So as mentioned, one of the major challenges, the data that's being produced in the genomics and epigenomic field has really been exploding. There's ENCODE, there's GCGA, there's the Roadmap Epigenomics project. I'm actually part of this Roadmap Epigenomics project. I built genome browsers for them. So lots of the discussion that is going to be focused on examples or discussions is going to be focused on this project here. So this is putting one of the first so-called epigenome atlas data on UCSC genome browser. And then this is actually using some of the more advanced visual technology by overlapping multiple tracks in a same track space so that we can compress the data. So basically, if you look at a K4 monomethylation, this enhancer track, we can actually put multiple data in one space. So this is this rainbow display. So it's already getting pretty complicated, and this is another way of looking at the data on the UCSC genome browser. It's a simple question. We have a lot of data now. In the Roadmap project, we've generated over 2,000 genome-wide sequencing-based functional genomic data. ENCODE has more than 3,000, right? So this is really starting to test the limit of our genome browsers. So the first thing I want to discuss is can we make this like Google Map like for biologists so that you can navigate the data in a smoother way. So our attempt was this human epigenome browser at Washington University. So because the field has been growing so fast over the past couple of years, we were able to publish this work 2 years ago, and then again, another paper describing the update of this browser will come out in a month or two. So this is the website for this human epigenome browser. It's the main product of this project. So I'm going to give you a little tour of this browser, and later on, hopefully, we can do some real life online tutorial at the poster session. So the concept is very simple, right? When we have lots of data, when we have a limited computer screen to look at the data, let's compress them. Let's use heat maps instead of those wiggle tracks. So the one very simple, I don't even want to call that innovation because people have been using this all the time, is to encode your data in this heat map version, heat map format. But the real innovation is to put this metadata heat map on the side, right? So this is a colorful way of encoding the information of your data, your sample, the age, gender, response to chemo, whatever you have as a metadata for the data. When your data is like hundreds and thousands, to that range, you need that metadata to help you organize your genomic data. So that's probably one of the earlier innovations for this line of work. On the technical side, we have we don't put this data in database, right? So they stay as a multi-resolution data format, and lots of the imaging rendering are really all done on the client side to avoid the traffic invoked with sort of displaying large amount of data. So these are the chromosome coordinates. These are experiment tracks. So this is what we call genome heat map. And over here, we have, again, the metadata heat map. And then the implementation allows us to do lots of, some of the Google map, like drag and move, pan zoom, sort and order. So you can drag on the heat map side. You can drag on the sort of a metadata side. You can drag on the genomic data side. So we're trying to achieve a very smooth sort of visualization experience for the investigators. And of course, the typical zoom in, zoom out are also we make that sort of Google Map-like and smooth. So when you zoom in to the sort of a very high resolution, you can see the genomic sequences, and these are sort of the genomic data at sort of a higher resolution. And you can also look at sequence rates that align to the genome, and then look at the variations in each of the rates actually align to the genome. So there's multiple levels of resolution encoded in this data. And lots of sort of a user control. So basically, this part is really give the investigators full control of this tool. You can do whatever color you want. You can change the height, different threshold. You can by changing the threshold, you sort of in a way get around this peak finding problem, right? So instead of showing the peaks, you show the raw data. You change the threshold, you essentially define your own peaks. So this is sort of the data at different threshold. You can change the colors of your data, the threshold of the data. You can turn heat map into this more conventional wiggle plot to focus on a smaller set of the data. So basically, this we try to give the investigators as much freedom as possible when they manipulate this data visualization. Each individual track will give you, by just right click on the tracks, it will bring out sort of information control panels. Get you the geo record, for example, the information, how the experiment was done, what kind of processing has been applied to this particular data, what is the mapper that you used, what are the read lengths of the data, and so on and so forth. And then you can also look at a genome-wide snapshot of your data, right? How does this data look like globally from like a bird eye view? Okay, so basically, this is this is just a collection of tools that are that gives investigators as much flexibility as possible to explore and look at the data. And one of the key advantages is we can now visualize hundreds of tracks on the same computer screen. Okay, so so that's on the sort of the user experience, but more and more importantly, people want to not only look at the data, but also do some bioinformatics with the data, especially for biologists. I mean, Esther showed the very nice figure by overlapping different peaks, and get a nice profile of peaks. So you still need certain expertise in bioinformatics to be able to pull something out, put something like that out, right? So our goal is to make this genome browser a bioinformatics engine, so that so biologists, without having to code anything, you can generate those figures by just clicking a few buttons, right? So first of all, you can upload your own data. You can put your own data on the browser. In fact, on the technical side, none of the data actually got, so very few, a small proportion of the data get actually transferred. Most of the data actually stay at your sort of a local disk. And then using this example, so let's say we can collect a bunch of genes of interest, right? And then we'll, instead of looking at the entire genome or in one gene at a time, let's just look at the genes that we're interested in, right? So this is how the browser look like under gene set view. So these are individual genes that you're interested in, right? Now, this is actually a very, very simple principle. Couldn't be simpler, right? Genome is linear. There's a coordinate system. What we're doing here is just to break that coordinate system. We can bring different parts together. This example shows that we can bring some genes together, right? As you can imagine, you can bring other things together. You can bring a bunch of promoter together. You can bring a bunch of enhancers together. Now, so the only innovation, if you will, is a simple principle. Couldn't be simpler. That is breaking the genome coordinates, which is linear, a very nice coordinate, move them apart so that you can bring interesting parts together, right? So we implemented that, and with that, we can do a lot of damages to the data. So now this is the example where you bring your genes together, whatever gene of interest, you can bring them together now with a couple of browser clicks. And this is to show you can actually also select the different variants of that gene. And then you can generate some analysis, right? You take a data set, being methylation or histone modification, and you can plot the profile across different type of either across the genes, or you can summarize them on promoters, UTRs, exons, introns. So you can do a lot of this type of analysis. What? 5 minutes? Sorry. Okay. Yeah. Well, obviously this wasn't well planned. I'm not even halfway through. The point is you can get a peak, right? This is a peak for unmethylated transcription start site. So you can do this with just moving things around on the browser. Okay, I'm gonna quickly skip this. You can do different type of profilings. You can do clusterings by just again a few browser hit, right? So this again shows the unmethylated pattern of the transcription start site. Okay, so I'm gonna. I have a few examples, but I'm going to skip them. So this is an example showing bivalent domain. This is actually from Brad Bernstein's lab. This is showing 3 bivalent domains next to each other, all by just clicking a few browser buttons. This is actually one of my favorite examples, but I'm going to skip. This has a lot to do with my own research on transposable element. So this browser allows me to put all these transposable elements next to each other. Right? That's never been done before, at least in the browser space, and then show that there are transposable elements that are tissue-specifically methylated. Right? So that's a pretty cool discovery. And then there's sort of a, well, I guess I'll skip this. So I'll quickly go through this part, genome browser as a nonlinear system visualizing chromatin interaction data. So there are quite a few development already in the field looking at things sort of a high-C data, ChIA-pad data in the format of arcs and heat maps and circuits. So what we did is now just like where we can put different genes and promoters next to each other, the simple principle here is to bring the interaction partners together, right? So that's it. That's the end of the show, right? Really, we just come up with a way to put different interaction partners together. Again, breaking this linear system of the browser. So this is sort of the nutshell of the data. This is a region, right? And this is another region. They're 70 megabase apart in the genome. And the high-C data here suggests that this part of the genome interacted with this part of the genome, right? On a typical genome browser, you have to zoom out all the way and you lose everything, right? But by putting them next to each other, you can actually see this little cell that sort of sort of indicating the interaction between these two regions. So this is how we encode. So we have sort of a different ways of looking at interaction data. This is this arc view, sort of ChIA-pad data usually come in this way. So each arc connect two interaction partners, and this is the high-C data. So some of you probably seen this data before. This little cells, each little cell represents the likelihood of two regions interacting with each other. So the technically it's sort of sophisticated, but the concept is really simple, right? So breaking down the linear coordinates and move things apart, move things together and then all of a sudden we have a way of looking at interacting regions. So I'm going to skip the details. You can look, there's lots of details hidden in this data. You can look at individual arcs and look at the genomic regions that are connected by these interactions. And you can bring another browser panel out, right? So basically, you have a browser in the background, you have another browser jumping out, and then you can look at this is where your browser is, the current locus is interacting with sort of on a remote site, right? Skipping ahead, this is really what we can achieve. We can have multiple panels like this, on top of your genome browser, giving you full control of the genomic data, epigenomic data behind the scene, behind that region, and perhaps you can start, as an investigator, you can start to explore why these regions are interacting, right? And then obviously another very nice view, first invented by the Circus group, to look at chromosomal or genome-wide interaction. So we can put everything together in this sort of a browser context. Right? So this is just a view different ways of viewing the data. And in addition to this, so this shows this location is interacting with all the other locations on this chromosome. And on top of that, we can now decorating this circlet with the genomic data, RNA expression data or histomodification data, DNA methylation data, so allow you to get a sort of full picture of the data and then also allow you to jump in and zoom in to specific parts and look at the interaction and then look at the epigenomic data. Skipping this, skipping ahead. Finally, genome browsers are becoming a community resource. It's becoming more and more difficult for one single group to collect all the resources, collect all the data, and then present to the community. So we're proposing this browser hub model. So this is a model that, in collaboration with UC Santa Cruz, we initially developed for the roadmap epigenomic data. In a nutshell, is the visualization is sort of centralized, but the data is distributed. Right? The data is distributed. You don't need to transfer the data or the entire data set to the site of visualization, but you can achieve sort of integration, sort of a virtual integration of the data. So, whoops. So something's didn't show up. So this is sort of a genome browser view, if you take my word for it, from UCIC. So you can actually see the data physically located in St. Louis and data physically located in Santa Cruz, from the same browser view. As an investigator, you don't have to worry about where the data is located, right? So we have implemented the same concept in the UCIC in the WashU epigenome browser, where the hub function and the capstone track function are sort of working this way. And now we can put all the roadmap data, the long-range interaction data, ENCODE data, all together under this one umbrella, but as resources distributed out there in the community. Okay, so the last site is sort of an effort to bring out all the complete epigenomes sort of produced by the roadmap epigenomic community. Now this is again, this is a very simple innovation. That is, we allow multiple browser instances to be displayed and navigated in the same view. So at the end of the day, this is what we have built, right? So this is the matrix that we want to be able to navigate with the browser systems, with the genome, with the different types of data, with different types of samples. And we're trying to figure out a way so that investigators can jump to the regions of interest quickly, and then also be able to sort of zoom out and look at the global feature of the data. Okay, so that's the end of the talk. I'd like to acknowledge, and lots of the earlier work was initiated when I was a postdoc with David Hausler at UC Santa Cruz. Lots of the work we're doing is in the context of the roadmap epigenomics project in close collaboration with Joe Castello at UC San Francisco. And these are the folks who generated this next generation long-range chromatin interaction data that recently populated our epigenome browser. And this is the postdoc who did all this work. All right. His name is Xin, and again, he has a poster later. Thank you.